Hello again. This is the continuation of some lecture material regarding the cell. This is all part of the first week of this lecture course. Again, we have an alternating format between the sort of the biological and the more radiological. So, and I'm, of course, Dr. Stein doing the biology portion of the program. Nevertheless, this is kind of where we left off. You'll see this as power. This is, again, PowerPoint B. We also are going to be doing D, which, depending on the timing, we may get through both of these simultaneously today to conclude week one. Not necessarily so, but I will, I beg your pardon, start on with this. So, nevertheless, we were talking about means of transportation across living membranes the last time. And this is sort of the area where I'd like to continue. So these are the active transport. We dealt with the effectively passive transport, diffusion, facilitated diffusion, and osmosis. These are active transport mechanisms. And by definition, active transport mechanism, whether it's active or, as you can see here, vesicular, requires energy. And so active transport is effectively, the best way to describe it is moving against the concentration gradient, or in certain circumstances where you have to impart extra energy to move various things in various directions. Vesicular transport would best be described as bulk transport. It's sort of the way a living cell will effectively take a truckload. It'll either deliver a truckload, that's exocytosis, or receive a truckload by endocytosis. And as you'll see, subsequently, endocytosis involves 3 different subvariants called phagocytosis for solids, pinocytosis for liquids and oils, and receptor-mediated endocytosis, which even though you might categorize as a third type, is not necessarily a third type. It's maybe a subset of the first two, phago and pinocytoses. So the whole idea is when you're doing these things, you need energy. It's too large. It can't get through the membrane because it's not compatible with fats. It's not lipid soluble, and it's not about the concentration gradient or going against it. And because we're expending energy from a cellular basis, we need to be able to move other things along with it. So we can either use the current or the flow of things moving in to bring things in. We call that sim or co-transport. And by the same token, we can use that opening and some of the energy that's involved and transport more than one thing out, or do things, open one door in one direction and another door in a different direction. That's called antiporting, and it's used all over the human body for a variety of cells. Okay, and again, the energy requirement is implicit in this particular discussion. So we have primary and secondary active transport. Primary is what you're moving by expending the ATP. Anything secondary is bringing something along, using that energy and indirectly opening that other door to move things in the opposite direction. So, and that's the distinction. I don't believe that that's terribly a big deal. Classically, you will see, as we look more at cell functionality, that primary active transport is basically utilized, and particularly so by something called sodium potassium ATPase, or sodium potassium gates. And what they do, I beg your pardon, sodium potassium pumps. And what they do is they're constantly resetting the cell's electrical activity, which you will see shortly, subsequently, without going into a great deal of detail. It's really not germane to this course. For many of you who had an introductory biology course or anatomy and physiology, depending on what the requirements are for your particular field of endeavor, that's, basically you probably were acquainted with some of that. So you can see them here. So shape changes effectively facilitated by ATP that allow things to move. And again, we are seeing a lot of these different things. We have calcium pumps in the muscle, for instance, proton pumps in the gut to create the acidity of the gut and the stomach. And the famous and the most abundant of all these sodium potassium pumps, and their whole job is to reset nerve and muscle cells, which are more or less effectively constantly firing. And that's where the vast majority of our energy, more than half of our energy resources in the form of ATP, go to these pumps. Okay. Here it is, the most studied one, pumps in and out of the cell, all plasma membranes. And even though all membranes are constantly needing tiny adjustments in what we call the resting membrane potential, that's not the charge in the cell or out of the cell. That's the charge right across the membrane. By definition, living membranes are polarized. They're a little bit more positive on the outside and a little bit more negative on the inside. And that's a really significant factor when those nerves are, when nerves and muscles are excitable. That excitability relies on that being present and can be altered substantially if it is not. So effectively, we see a variety of different channels that are involved. Okay. And so this is sort of the background of why those sodium potassium pumps are so important and why they're essential for the functioning. We have openings called leak channels. They require no energy because there's always more sodium inside this. There's always more sodium outside the cell and more potassium inside of the cell. And so they're constantly little bits of sodium leaking in, little bits of potassium leaking out. There's a lot more potassium, as you will see subsequently. And because of that, a lot more potassium leak channels, the cells tend to push out more of this potassium to the point where it makes it quite a negative environment right under the membrane. And that attracts back some of the sodium, and that creates something we call the resting membrane potential, which is about minus 70 millivolts when you measure it in living cells that are there. And effectively, because you're getting that constant flow of sodium in and potassium out, you have to basically redirect it because you're altering the internal contents of the cell. So every time they're to make subtle adjustments in a cell that isn't excitable, like a nerve or a muscle cell, there's always a little bit of activity. And typically the activity, as you will see through a series of these slides, without going through them, is a 3-to-2 relationship. Again, sodium is a smaller molecule than potassium. The pumps basically use the same channel passageway. Sodium and this is charged sodium, not metallic sodium or metallic potassium, which they are metals. If you had a big lump of them, they'd be sort of like a soft metal, almost like a chalky kind of consistency if you could expose them to the air because they're in oil. Effectively, you can take 3 tiny molecules of this charged particle sodium and move them in, but you only have room for 2 potassiums to be spit out. And that's a pretty consistent relationship when we look at nerve and muscle physiology. That 3-to-2 relationship, it demonstrates it here. As you can see, 3 sodiums bind to the pump, we get some ATP. Okay, that when we utilize the ATP and release the phosphate, that's called phosphorylation, changes shape. Basically, now it spits the sodium back outside and simultaneously or shortly thereafter brings in 2 potassiums. And again, but some additional phosphate is used and brings those back in, and voila. And this is an illustration that really dictates it. It's available to you. It takes maybe 2 or 3 minutes to listen to. Basically, as you will see in some of the PowerPoint sequences, these particular animations are useful. It gives you an idea very much of how significant. I'll buzz through it, meaning I'll play it. I'm hopeful. There it is. And there is narration with it if you enable that, and I have the volume.
The human brain alone contains.
So as you can see, this is a very good construct of, beg your pardon, what the membrane looks like. Outside. So there's a lot of these channels that are there, and you can see in the purplish ones versus the orange ones are different. You have more potassium and sodium. Outside. And so I would encourage you to listen. But this is what happens. It's directly across the membrane. Again, the interior of the cell and outside the cell and the interstitial or extracellular fluid is got no or doesn't have any appreciable charge. And so they're showing you sodium leaking out and potassium leaking in. It's because there's bigger part sodium leaking in and potassium leaking out. There's always more sodium outside the cell. There's always more potassium inside the cell. And that's where the pumps come in to constantly maintain that concentration gradient. And if you go through it, what it'll show you is that as the potassium leaks out, okay, again, it creates about a minus 90 millivolt, and it attracts a little extra sodium to come back in to balance it out, and sort of stabilize it minus 70. Then everything else is more or less equilibrium. And what equilibrium just means simply is that you're going to have a few sodiums coming in because there's a gradient, and a few potassiums leaving out because there's a gradient. You have those pumps constantly working to make sure it stays there. And so depending on what of these different primary transport system is utilized, okay, and so the sodium potassium pump is well expressed to strengthen it. It basically keeps the concentration gradient, which is the force that used it. And absolutely, that energy can be utilized to have other what we call symporters or co-transports to bring things in. A lot of times we're moving sodium into a cell, we're moving glucose into a cell. It helps that that's kind of the way insulin works. And so they're demonstrating it to you because that little hexagonal, and inevitably when you see these hexagonal, they imply it's a simple sugar or a monosaccharide, and the ones we utilize the most in our bodies are hexoses, 6 carbon sugars. So each of those little points represents a carbon. And that you see that all the time, whether you're in a textbook or an animation. That's a pretty consistent way of demonstrating it. So in it comes, out it goes, etc. And so the drive from this is helping the drive from that. That's there, and they're demonstrating it to you. What we do a lot of extensively in our cells is vesicular transport. It's principally the way our cells eat and drink and how they basically excrete wastes. Okay, it's also the way that at the neuromuscular junction or nerve to nerve, we excrete neurotransmitters. It's kind of the way we do business. When we bring large items into the cell, or we bring a large amount at the same time, inevitably, we have to utilize some type of vesicular transport. What's a vesicle? A vesicle is a membrane. Basically, it's a circular, it's like, think of it, if you two-dimensionally as a circle, three-dimensionally as a sphere. And it's a little container made of membranes, good old phospholipid bilayers with embedded proteins that have the remarkable ability to either develop from the cell's membrane or fused to the cell's membrane and bring in or spit out. So they either envelop something and bring it in, or they create a membrane around something they're going to spit out, send that over in close proximity and join it to the cell's outer or cell membrane and basically spit it out. And in order to do this, you're constantly using energy. It's another large energy drag, so to speak. It's another way that we have to spend a lot of our ATP. And here are the different types I talked about. And again, frequently phagocytosis and pinocytosis utilize different kinds of receptor. The one receptor-mediated endocytosis is very specific, and it involves fatty substances. And that's a very well understood particular mechanism. So there's effectively three different kinds of ways in and effectively one way out. So whether you're excreting a neurotransmitter or a waste product or any undesirable residue, that's exocytosis. Transcytosis is interesting because it utilizes a membrane, and we do this a lot in digestion. We do it a little bit in the kidney, and what it does is it's basically bringing something through the cell. The cell's just a passageway, and that little vesicle, that little membrane container, makes its way through the cell without interacting with what's inside, and that's very useful. Because a lot of times we're bringing in things that cell doesn't have to deal with. We're going to deal with it someone else in another organ or another location. And so really it's more about just encasing it for transport. Okay. And so that's where we talk about vesicular trafficking. That's moving it from one area of the cell to another to be able to modify it. And you mostly learned about that. It's like taking stuff out of the smooth endoplasmic reticulum and going to the Golgi apparatus, and that's where modifications occur. It's like the packing and shipping department. Okay, in your cells inside, playing around with the materials that are there, making them more compatible. It's just like buying something on the internet, and you hope they pack it properly because you hate to order something that's fragile and it comes in broken. Would be the way to look at that. Okay, endocytosis, bringing it in, formation of, and these are, and again, this is where the receptor mediated kind of overlaps. Most of the vesicles have different kinds of proteins in them anyway, and they function as receptors. So stuff comes along close to the cell, it triggers those receptors, and it causes basically a subtle shape change in the cell to start to envelop the particle. Okay, so the substance being pulled in has to be able to effectively bind to the receptors. And you'll see the difference with receptor mediated. Here's a big problem. Bad guys, toxins, bacteria, viruses can mimic that. They can effectively adhere to it. Surely the most famous of these is HIV, and in human immunodeficiency virus, it binds to the receptor called a CD4 receptor, and you may have learned about that or not. That's the receptor on a white blood cell called your helper T cell, which is the single most important cell in your immune system. And basically, it uses that as a doorway, an entry point to get inside and infect and damage that cell line. And if it's not treated, it's going to lead to death. You're going to develop AIDS and you're going to die. And so once the vesicle enters, if it's going to be processed in the cell, it fuses with something called a lysosome. That's an internal organ that's used for digestion and defense. It has enzymes that'll either gobble up and process and utilize the material, or they'll destroy it if indeed it is some kind of a bad guy. The HIV is not a great example of that because there's some things that it almost overwhelms the system. If you get a lot of bacteria or a lot of viruses that come in, okay, your mechanism to deal with them becomes limited, and eventually one of them becomes fixed, and now you have a big problem. And again, if it's just going to use the cell as a passageway, getting from, like, say, your GI tract into your bloodstream, we call that transcytosis. So the names you're going to see. So, and this is a classic picture. Here it is. Effectively, the protein coats are there functioning as receptors. The stuff comes in, and what happens after it uncoats with those proteins, not a big deal for us, it fuses with a lysosome. So if it's just being transported, it comes in, it doesn't fuse with the lysosome, it just gets spit out. And it doesn't depict that quite as well as some other illustrations do. But in this particular instance, it fuses with the lysosome. The enzymes in the lysosome either, again, digest or destroy, in some way process it. If they're going to use it, it's released into the cell. If there's waste material, it's basically tossed out of the cell by exocytosis. And they're just showing you sort of little numerical creations there to demonstrate this. So the different kind, that's generic phagocytosis, and the way this happens famously is called pseudopods. If you ever looked at pond water and saw an amoeba, it's like a baby scooching on the floor. It can sort of change shape, and what it does is tiny filaments push on your cell's membrane and creates sort of a change in their shape so they surround something and eventually engulf it and effectively pull it back in. And that membrane that's surrounding it becomes effectively the membrane of the vesicle that's there. And so the vesicle is then called a phagosome because it gobbles something up. And this is a classic way your white blood cells work. Very much so macrophages, which is sort of your body's garbage disposal system. You know, they travel along and find debris and gobble it up and get rid of it. So again, the amoeboid motion, these are, and it's well stated to be temporary extensions. So they don't stay there. They're just used for that process when they're attracted to or triggered by a chemical striking a receptor. And so here it is. Here are the receptors. The receptors will target this material or adhere to the material, and it will induce these little changes in the shape from being flat, let's say here, to causing and these are what pseudopods mean, false feet. and they engulf the particle, and eventually it becomes surrounded. Same thing for penocytosis, cell drinking, but really it's when they're not quite solid, but they're more in a fluid or oil state, dissolved, etc. And then they also become an endosome. And we see a lot of this in different environmental samplings. Interesting. It's one of those interesting things that our immune system is always doing in various areas of your body. They're sampling stuff that comes in to see generically if it's something we should be responding to or not. And look, you and I, every time we take a breath, we're inhaling bacteria and viruses and particles and materials. We can't help it. Air's not sterile. So, and if it was, it wouldn't be beneficial to us because we need a lot of the things that are in there, particularly the oxygen. But of course, we have to have this. I always think of it as sort of taking your immune system and keeping it in tune. It's constant. It's like in the winter, not starting your car all winter and you come out and, oh my God, I can't start it. You have to constantly use it. And a lot of this blows back in medicine and science and disease to something called the hygiene hypothesis, which means that when little kids are not exposed to things, a lot of times, if their first exposure after being sequestered, okay, their moms and dads don't let them play with other little kids, and they keep the place ridiculously clean and don't go outside. I heard little Johnny had a cold last week. I don't want you playing with him. You're not really doing your kid a service. I mean, their body has to be exposed to this thing and respond to it. And historically, this is what they did with royalties, so the king's children wouldn't get sick. And what happens is they die prematurely because they can't handle it. Okay. So hopefully your moms and dads were like mine and basically shoved you out of the house and said, go out and play for a couple hours. I want to see you. You know, it's a Saturday. Don't sit in the house. It's beautiful and sunny. And, you know, that's what we did. So effectively, it's an interesting way to look at it. And it's really relevant today in the COVID era because that was, I understand, not to be critical of the restrictions we saw under COVID, but frankly, it was an over response. It altered a generation. And so if you're part of that generation that had to go to school from home and online and didn't get the interactions and didn't get the socialization and all the fun stuff that school was about, going to the game and hanging out and, you know, walking around the hallways with your friends and you know, having a good time and lunch, whatever it is, whatever you folks do, because I'm a long way detached from that, very much. But I think it really impacts everybody. So moving on. So nutrition absorption occurs. Membrane, and the interesting thing about membrane, it's all recycled. It's not like, oh, you use that membrane, the cell's going to shrink after it has a big meal. Remember, it's still spitting out stuff at the end. So we call it membrane conservation. So membrane that we sacrifice to bring something in, we're going to restore that membrane with what we spit out. So the cell effectively maintains its size over time. It may make it a little smaller after a big meal, sure. Okay, when is it larger? When it has to spit out a lot of waste, sure. But eventually, it's going to come back, and there's a penocytosis nugget. Receptor mediated. It depends on the molecule, and typically we see this what are called clathrin-coated pits. Clathrin is a very interesting compound. Okay, very, and it really makes it less generic and more specific would be the way to look at it, even though I think it's very similar to endocytosis. And what we see brought in, look at the things. Enzymes that your cell may need. This is really the most startling one, the LDLs, low density lipoproteins, sometimes erroneously called bad cholesterol. Low density fats are something that we have to utilize efficiently. And if we do not, and there are excessive amounts in the bloodstream, whenever there's damage to a small blood vessel, like a capillary or an arterial, they can accumulate there. And that's when we hear about people with heart disease or kidney disease. That's part of the problem. Okay. So it's one of those things that, and I'll talk about some of the strategies in a bit. So iron, which is something we obviously have to have. Insulin, which is such a extraordinarily important compound that, if we can't regulate that, you're looking at diabetes, and then bad guys, viruses, diphtheria, and cholera are toxins. So there's good and bad, and so these are interesting mechanisms. Okay. This is called caveolae. It's a fancy name. You don't have to know. Has smaller pits and different protein code from clathrin, but still very specific. And you can see tetanus is in there. You can see folic acid, which we have to have to appropriately make DNA and RNA, and tetanus, which will kill you. So here's just a generic example. You don't have to know a little bit about those specifics, and it's just for context. So here's the clathrin, kind of this funny looking shaped receptor. Here are the molecules, and when enough of them bond, then we effectively we create it. I'll flip back. Why the importance? The best one to underscore is these are your development of these, and specifically, let's say for low density lipoproteins, is hereditary. If you are genetically have a genetic tendency to not make these or make these sufficiently, it impairs your body's ability to bring in those fats and process them inside the cell to be useful. So there's more in the bloodstream. So when you hear about somebody, typically someone who's older, certainly middle age or above, has to take quote unquote cholesterol medicine, a lot of times people with those kinds of problems we call hyperlipidemia, too much fats and cholesterol in the bloodstream, what they have to do is to compensate for this. You have to find other mechanisms to get them into the cell, or at the very least, if there's excess, to render it so it doesn't help you to remove it. So that's one. It's not all about your diet, and it's not all about exercise. Some people can exercise excessively and be very cautious of your diet. Hereditary tendencies supersede that. And you're going to, if you don't know people, you're going to know people who do everything right. And unfortunately, because of this genetic defect, they have to take different medications just to maintain their health. And as long as they do so, they're probably going to be okay. Exocytosis is very simple. It's just basically spitting it out. We call them secretory vesicles. It's interesting because those vesicles, those little membrane containers, have tiny little proteins that, and they call them V snares because of how they work. And they have T snares on the membrane, their target, or wherever they're going, if they're going from one organelle to another. And in particular, when the vesicles are getting close to the cell membrane to be spit out or exocytosed, these snare proteins are enabled. And what they do is they lock onto it. And it's how neurotransmitters and mucus and waste products all go. And they show you a real interesting picture. If you go through it, here are the snares, V on the vesicle, T on the surface you're transporting. They fuse or interlock. That's why they call them snares. It's sort of like they grasp together. They fuse because it's really nothing more than just additional membrane, just like this membrane. And when they fuse and open up, out it goes. And here it is graphically in a micrograph, spitting out whatever neurotransmitter, let's say, acetylcholine. And they just show it to you individually like that. Again, sort of the nuance that you must have learned about, or maybe you didn't, that's associated with that, before I get back to resting membrane potential, is that's the mechanism that's basically blocked by Botox at the neuromuscular junction, so it can't spit out, if this was acetylcholine, and it basically selectively paralyzes muscles. That's what Botox is called flaccid paralysis. Basically, the muscles can't get any signals, so they start to basically they lose the ability to contract. And flaccid means they're not in a spasm. Tetanus is a spastic paralysis. And so here's a nugget about resting membrane potential. And again, it's separating the charge. Anytime you have a charge different across a structure, you create a degree of polarity, and we refer to that polarity as membrane potential. And how nerve and muscle cells fire is they rapidly reverse that potential, and it creates almost like a wave along the membrane that effectively will generate activity, whether it's movement, whether it's spitting out substances, whatever it particularly does. And so the resting membrane, and this is more potassium out, more than sodium coming in because of those leak channels, and a fancy term we call it electrochemical gradient. And if you scroll through this, you're going to see that right across the membrane is on average most cells minus 70 millivolts. What's interesting is that the interior of the cell and the exterior, as you get away from the membrane, is neutral. Chloride balances out on the outside. Proteins that are so big they can't get out of the cell balance it out on the inside, in the cytoplasm. And proteins in general, the ones that are exposed to the cytoplasm, their exterior, are generally ones that release protons so effectively they have a negative charge. And that's the offset. So you're going to see, depending on courses you're taking, chloride plays a role, but only if we selectively move channels for it. So right now it's not part of the resting membrane potential. And they're just showing you these are the proteins that are so big they're never going to get through there, and they offset all the positives here. So it's neutral here, and the chloride offset it over here. And then it's maintained. There's a sodium potassium pump. The last little nuggets of this first presentation I'm going to do, or rather the second of these three presentations from this PowerPoint sequence, how they attach to one another, these adhesion molecules, how they anchor cells, okay, and they play a role. All these are sugars that are attached to proteins, and again, those sugars allow other things to adhere to them because they're sticky, okay. So the cell, and again, cells other than white blood cells don't function as a unit. They're all attached. So they can be attached to what's outside. They can be attached to other cells. They have the ability for certain kinds of cells to move past one another that are there. When chemicals are released and trigger those kind of sticky molecules areas and alter them slightly, then they're going to hold on to white blood cells. A cell has no way it doesn't have vision, it doesn't have hearing, it doesn't have smell. So it's basically a lot of cells are moved by chemical attractants or repellents that are there. And so the cells are, their ability to move, and typically, again, it's amoeboid motion, is triggered by dissolved chemicals going through the liquid that the cells are surrounded by, what we call extracellular fluid, okay, or interstitial fluid, means between tissue fluid. And white blood cells are effectively driven as those contact receptors to sort of scooch over toward those areas because that's where an infection is or that's where an injury is. Okay, and lots of other things that they do that are there. And cells are very sensitive to that. And again, those glycoproteins are all about developing immunity, where groups of cells form particular organs, or our body gets to know what's supposed to be there, they're not. And then there's a lot of signaling. Some of those chemicals released during inflammation have a fancy name. We call them ligands that are there. And they go through a lot of detail about this. And they talk about different ways. And this is really not for our course. It just basically depends whether they strike on the outside and trigger a chain reaction on the inside. Insulin does that to a certain extent. Okay, epinephrine does that in fight or flight response. When we release that, it can't pass through the membrane, so it triggers a receptor, and that receptor sets off a chain reaction along the membrane, and all of a sudden, lots of different things happen inside the cell. We call it a cascade. Where if it's fat soluble, it works differently. It actually gets through the membrane, talks receptors in there. So I'm going to conclude this. I'll just give you a very brief introduction, pardon me, on what we're going to look at next, which is this. And this is something you've all heard before. I'm going to reiterate it. It's called the cell cycle. And as we scroll down it, I want you to understand, and again, I'm going to put some questions for each of these sequences in that you can look at almost as review questions you should answer. Probably put them in a multiple choice format. And so what you're going to see is this. We're going to look at that part of the cell cycle really a lot to understand what's going on. Okay. Okay. And why it's so darn important, and when we interrupt it, or particularly in the context of radiation biology, what it can do to that cell cycle, and why so many problems can be created, particularly with cell destruction and cancer. So I'll leave it to you at that. And I will get back and do another one of these either another time today or later as time permits. Thank you.